Hello, my name is Destiny, and today I will be going over two types of cell reproduction, mitosis and meiosis. I am going to start with talking about mitosis. As you can see, mitosis is at the top half, and meiosis is at the bottom. So, mitosis, you're going to start with a diploid number of chromosomes, which is represented by the 2N. In humans, there are 46 chromosomes present. We get 23 chromosomes from our mother and 23 chromosomes from our father that equal to 46. And in this, you see the arrows, which mean that the cell has gone through mitosis and has created two new identical diploid cells. Each of these cells also contain 46 chromosomes as well. And each of these cells can go through mitosis again. And this is how multi-celled organisms occur, because this cell can make two more and this cell can make two more, so it can continue endlessly. And in meiosis, which is at the bottom, we begin with the same diploid cell as we see in mitosis, but it goes through two steps. So the diploid cell goes through meiosis 1, and then we see it go through meiosis 2, resulting in our 4 haploid cells. The type of cells produced in meiosis are the sex cells, which are called the gamete cells. And the cells produced in mitosis is every other cell in the body. You are now looking at two cells. The cell on the top will again be a mitosis process, and the cell on the bottom will be the meiosis process. This will happen throughout the video so that you don't confuse each phase. So I'll separate it so that you can see it more clearly. Both of these phases take place before mitosis or meiosis occurs, and that is interphase. So interphase is the beginning process, and interphase has 3 steps. So it has the G1 phase, the S phase, which is what I'm going to be focusing on, and the G2 phase. So we'll have the G1 phase. The cells grow to almost its capacity. So it's almost its full capacity. And then the S phase, the DNA strands will replicate. And then the G2 phase, the cell will continue to grow. So as you can see, both of these cells, we are beginning with 4 DNA strands in each cell. So this is a similarity. Both mitosis and meiosis start off the exact same way. So we both begin with 4 at the top cell here, and then 4 DNA strands at the bottom cell right here. So now I'm going to focus on the S phase. So during the S phase, the DNA strands duplicate. So for both mitosis and meiosis, you will get twice as many DNA strands in the cell. So up at the top cell, you see that there's 8 now, and at the bottom, there are 8 DNA strands as well. We are now looking at the prophase stage of mitosis and meiosis. And notice that in meiosis, it is called prophase 1. And this, again, is because meiosis has a second round. So to differentiate between the first step of meiosis and the second step, we put a 1 on each stage. You can also notice that I didn't draw the full cell. This is just to allow more space for me to draw. So just keep in mind that this is all occurs inside the cell, not just in thin air. So in this prophase stage, you can see that the nucleus has dissolved. The next step of prophase is the chromatin strands have coiled into chromosomes. So you can see now we have the shape that we all know as chromosomes present. So for each of these chromosomes, you can notice that I have put an N1, a P1, an M2, and a P2. The M's just signal the chromosomes that are maternal, and the P's signal that the chromosomes are paternal. So M means that it's from the mother, and P means that it's from the father. And you can also see that I have drawn spindle fibers in the corner of each of the phases. The spindle fibers are made up of microtubules. These clusters of microtubules are responsible for moving chromosomes during cell division, and they are created during prophase. But these spindle fibers are very important in our metaphase and anaphase stages. And this would be the end of the prophase stage of mitosis. So this is where we're going to leave off, and now we're going to focus on the next parts of prophase 1 of meiosis. So now we're looking at the rest of meiosis prophase 1 stage. So during this stage, synapsis occurs with the chromosomes. This means that the homologous chromosomes become pairs, and each of these pairs are called tetrads. In each pair, there are 4 chromatids, so each is a tetrad, and that is where the name derives from. Then we can see that crossing over then occurs. This means that parts of the maternal and paternal chromosomes swap with one another. It can also be called recombination, but crossing over is a more popular name for the process. This crossing over stage is why no two siblings are similar except for twins that are identical. It switches up the gene pool so that everyone is diverse and different. The formation of tetrads and crossing over is one of the biggest differences between mitosis and meiosis. We are now looking at mitosis and metaphase. The sister chromatids are now randomly pulled to the middle of the cell by these spindle fibers. A similar process occurs in meiosis, except for the tetrads are randomly pulled to the middle of the cell by the spindle fibers. The spindle fibers have this ability of movement because the microtubules attach to a protein structure called connectochores. The connectochores are attached to the sister chromatids, so that allows for the microtubules to move the chromosomes around the cell. And now we're looking at mitosis anaphase. Chromatids are pulled to the opposite side of the cell by the shortening of the microtubules. Again, meiosis anaphase 1 is very similar to mitosis, in which the tetrads are pulled apart to the opposite sides of this cell, and the tetrads break into chromosomes. And we are now looking at mitosis telophase. The nucleus then regrows around the chromatids. The chromatids then unwind back into chromatin. And lastly, cytokinesis splits the cell in half. And this creates 2 identical diploid cells. And this is the end of mitosis. So we are now looking at round 2 of meiosis. Mitosis has already ended, and we do not have a interphase for meiosis. For meiosis 2, we jump straight into prophase 2. So in prophase 2, the nucleus redissolves, and we again see the spindle fibers beginning to form. In metaphase 2, the chromosomes are again pulled to the middle of the cell by these spindle fibers. In anaphase 2, the chromosomes are finally pulled apart and the chromatids move to the opposite sides of the cell. Be aware that during meiosis 2, we are looking at two cells throughout. And this is represented by the dashed line. So it goes into each corner of the two cells and preparing to split into four cells. And we are now looking at meiosis telophase 2. The two cells, have been split into four. The nucleus reforms in all four cells, and the chromatids unwind back into chromatin. And then lastly, cytokinesis splits the cells in half again. So that results in four haploid cells. And now this is just a recap. Thank you for watching. I hope mitosis and meiosis is a little bit easier for you guys, and I hope I explained it well, and that you understand the similarities and differences between the two cell reproduction processes.